Hello, good morning. Good morning. Yeah, sound is clear. Yeah, okay. So today we talk about one important point, cytoskeleton. And under the cytoskeleton, we'll try to understand what is the exactly structure of centriole and what is the structure of cilia and the flagella, eukaryotic cilia and eukaryotic flagella. Because one important question asked from this point, that is 9 plus 0 arrangement and the 9 plus 2 arrangement. So we try to understand because थोड़ा सा complicated है students, so many times students feel some problem, specifically this problem, how to remember structure of centriole and how to remember structure of cilia and the flagella. Okay, so we are taking very important point or the very how to remember all those concepts. So we talk about एक yeah. Hmm. Chal. So today we talk about the functions of the microtubules, microfilament, and the intermediate filament. Last class we already started with a few concepts like microtubules. Basically, it is highest diameter 25 nanometer. Then microfilament, it has the diameter approximately 5 to 6 nanometer, and intermediate filament, that is between these two, it having approximately 10 nanometer diameter. And specifically, when we are talking about the function of the cytoskeleton, they are responsible for these are the supportive framework. Obviously they provide the mechanical support. Again, they responsible for the determination of the shape of the cell, as well as they are responsible, they help in motility also, like हम आगे देखेंगे. Microtubules, basically they responsible for the formation of cilia and the flagella. These are the major function. And another one, entire cytoskeleton structure, they made up by the protein, proteinaceous structure. प्लीज रिमेंबर, ना ही कार्बोहाइड्रेट है ना ही यहाँ पे कोई लिपिड का पार्ट है. एंटायर स्ट्रक्चर ऑफ साइटोस्केलेटन, दे आर प्रोटीनेशियस इन नेचर. So if you read NCERT, you will get this sentence, one question from this sentence. Okay, yeah. So next part we are talking about with respect to functions of microtubules. So first of all, we start with the microtubules, and normally we are talking about microtubules, that is MT microtubules. Number one function. After this chapter, we'll start with the cell cycle cell division, and during the cell cycle cell division, specifically we are talking about cytoplasmic division, that is called as the cytokinesis. Cytoplasmic division. So basically, microtubules, they help in cytokinesis of plant. They help during cytokinesis in plant. Number one function. Detail we'll discuss in next class, next chapter actually. Number second, basically, whenever we are talking about these microtubules, microtubules basically they are responsible for formation of, for the test only cell chapter. Entire cell, okay, entire cell. So we are talking about microtubules. Basically, they are responsible for formation of spindle fiber. And what is the spindle fiber? How that spindle fiber, it is play very important role during the chromatid pulling, etc., etc. All these function will discuss in chapter cell cycle cell division. Next, third point, microtubules basically they responsible for the cell movement. And how? Because you know very well, two important part. Number one, cilia, and another one, flagella. We are talking about eukaryotic cilia and the eukaryotic flagella. Okay, so cilia and the flagella, in case of the eukaryotic system, simply these are the made up by microtubules. So cilia as well as the flagella, in case of eukaryotic system, they made up by the microtubules, right? And the last one, we are talking about a simple process, that is intracellular transport. Check your mic. Who's that? Who's that? Check your mic. Dr. Suhas, check your mic. Hmm. So intracellular transport. What is meaning of the intracellular transport? Suppose these are the one cell. Inside the cell, now there is a presence of at the center. These are the nucleus. Now one vesicle they can move from periphery to center and center to periphery. So we are talking about what? Whatever the vesicles they can move inside the cell from periphery towards the nucleus, from nucleus towards the periphery. So such kind of the transport, simply it is called as the intracellular transport. Intracellular transport nothing but that is vesicle movement within the cell. Within cell. So you know very well, protein enzyme, they never release at it is, because inside the cytoplasm protein is present, inside the cytoplasm lipase enzymes are present, so they can degrade such parts. So that's why if you want to transport any material like protein, like any sugar part, any lipid part, that time you have to pack inside the vesicle, and now with the help of vesicle you can simply transport from one place to another place. So intracellular transport, vesicle movement inside the cell, all these are the carried out with the help of the microtubules. So these are the four important functions. Help in cytokinesis in specifically plant. Chromosome movement में help करेगा because they can form a spindle fiber. Again they are responsible for the cell movement because they play very important, they are the important part of cilia and the flagella structure, and as well as they are also help for intracellular transport, that is vesicle movement within a cell. Now next one, number one, we are talking about MF, functions of microfilament. Basically when we are talking about the microfilament, again they are responsible for the cell division, that is cytokinesis. They help in cytokinesis, but now in this condition we are taking cytokinesis in animal, because next chapter will discuss karyokinesis, that is nuclear division is approximately similar in case of plant and animal, but when cytoplasmic division, two different scenario will be follow. In case of the animal basically cleavage furrow technique used hota hai, and in case of the plant cell plate formation technique will be used. So in cell division point, that time also we discuss what is the exactly difference between cytokinesis in plant and animal. So please remember, microtubules help करेगा for the plant cytokinesis, microfilament they help for animal cytokinesis. So please don't get confused, one question from this point. Now number second function, basically they are also help in cyclosis, and you know very well cyclosis हम पहले discuss कर चुके. Cyclosis nothing but cytoplasmic streaming, that is movement of cytoplasm around the vacuole, maybe clockwise or maybe anti-clockwise. So such kind of this movement simply it is called as the cyclosis movement. Number third function, basically they support to the plasma membrane. They support to the plasma membrane. Normally what happen? Suppose the plasma membrane, these are the PM. Below the plasma membrane, now there is a microfilaments are present who provide a sheath, who provides the support. So now what happened? They form a one sheath-like structure. They form a sheath, and on the basis of that, simply we can say, sorry, excuse me. So simply we can say they support to the plasma membrane, because exactly below the plasma membrane they can form a sheath, and with the help of that sheath definitely, yeah, definitely they are responsible for, thank you bacha log. With the help of that sheath they can support. Now next one, number 4, now also they help in the movement, that is pseudopodial movement. Yes, can you tell me where the pseudopodial movement is present? They also help in pseudopodial movement. कहाँ पे present होता है pseudopodial? Pseudopodial movement also called as the amoeboid movement. Correct. So it is present in amoeba. Very good. So pseudopodial movement, nothing but that is the engulfment of food. And normally what happen with the help of the microfilament, they are showing extension. They can engulf the food, and normally they entered into digestive vacuole, they entered into food vacuole, and inside the food vacuole, proper food will be degraded. So pseudopodial movement में help करेगा, and pseudopodial movement also called as the amoeboid movement because specifically such movement present in amoeba. ठीक है? Yeah. And the last function, they are also helping endocytosis process. Last class हमने देख चुके हैं what is meaning of the endocytosis, that is engulfment of the food, formation of the vesicle, and if solid food, that time that vesicle called as the phagosome, and if liquid food, that time that vesicle called as the pinosome. So simply we are talking about which process? Endocytosis process, engulfment of the foreign particle, engulfment of the foreign food, and through the plasma membrane, and now finally there is a formation of a specific type of vesicle. ठीक है? Yeah. So any doubt from the microfilament function? Any doubt? Sandesh? Okay. Yeah, Chrisley. Now next point and the last one, intermediate filament. Last class also we discussed, means last class yesterday, huh? So basically that intermediate filament major function, they provide strength, they provide stability. So normally हमने देखा था keratin protein, basically these are one hair protein, because if keratin protein it is present in good amount, that time our hairs are healthy, they are very what we say, they are very strong, we are not facing any hair fall problem. But if the keratin concentration it is get declined, ultimately you can face different मतलब बहुत सारे reasons होते हैं for the hair fall. One of the out of that there is a one reason, concentration of the keratin. These are also. So next and the last one we are talking about number third, that is functions of intermediate filament. Number one, basically first function, they are responsible for the maintain the shape of the cell, maintain shape of the cell. Number second, basically they can hold the cell organelles. They can hold cell organelles. Number third, they also help in cell to cell junction. Cell to cell junction, and you know very well, cell to cell junction means, you know, in animal part, in zoology part, you will know very well, because बहुत सारे transporters होते हैं. Two cells they can communicate with each other through cell to cell junction. And next one, basically we are talking about the support strength to nucleus plasma membrane. And last class also we discussed one example. That example basically it is called as the lamine protein. Okay, example basically these are the lamine protein. So in genetics we'll discuss one example related to progeria. With if is this any mutation in lamine protein, so what happened, what kind of the changes will be occur, so that part means we'll discuss in 12th standard syllabus in inheritance chapter. Okay, yeah. So that is the simple straightforward function with respect to intermediate filament. So any doubt from the function? Any doubt? Because straightforward question तो नहीं बनता from the function, but such are the concept, they ultimately link with the cell cycle cell division chapter. Chalo, should I continue next part? Should I continue? So quickly we are taking one question which is related to microtubules. Microtubules, that is empty. Basically microtubules, simply they made up by the protein, and name of the protein that is tubulin protein. name of the protein, that is tubulin protein. Okay, basically two types of tubulin proteins are there. Number one, that is alpha tubulin. Number second, that is beta tubulin. Alpha tubulin protein, and the another one, that is beta tubulin protein. Normally what happen, if we are talking about these some functions related to microtubules, now they are responsible for the cell movement, now are responsible for the cytokinesis, etc. हमने बहुत सारे functions देखे. So now what happened, that time one tubulin they join with the another tubulin. So normally क्या होता है, suppose these are the tubulin alpha, beta, alpha, beta, alpha, beta. So normally what happen, one alpha tubulin, one beta, one alpha, one beta, one alpha, one beta. So normally like this, all these are the process, they are showing the elongation, they form a chain. All tubulin protein they join with each other, and now what happened, they are showing elongation. And please remember, for the elongation there is a requirement of energy in the form of GTP. So for money chahiye, and money in the form of GTP. Normally ATP, these are the energy currency. In maximum cell movement, in maximum cell activities, cell required ATP. But when we are talking about microtubules function, because see, single tubulin protein koi kaam nahi karega. We require whatever the tubulin proteins are present, that tubulin protein they join with each other, they are showing elongation, they are showing polymerization, and when all tubulin protein means alpha bind to beta, then alpha, beta, alpha, beta. So now they form a typical elongated chain. That time we can say for the elongation there is a requirement of money in the form of GTP. यहाँ पे ATP use नहीं हो रहा है. Please remember, ATP, these are the energy currency, definitely, which is involved in maximum reaction. But in this condition, for microtubules elongation, for microtubules polymerization, requirement of energy in the form of GTP. Got it? Clear this point? This point is clear, everyone? Yeah. G stand for the guanosine, baba. Guanine, guanine का होता है ना. So guanosine triphosphate. प्रत्युषा. ATGC वाले G है वो. It's a guanine, but when you become triphosphate, we have to say guanosine triphosphate. ठीक है. चलो. Yeah. So next part, ये था one question. Another point, one sentence it is given in NCERT. That is the boundary. The boundary of microtubules, MT. The boundary of the microtubules, they consist of total 13 protofilament. So what is meaning of protofilament? How many protein filaments? Total 13 protofilaments are there. So we'll try to understand. हाँ, they are helically arranged. वही diagram बात कर रहे कोमल, वही draw कर रहे हम. Yeah, कोमल. So we are talking about the boundary of the microtubules consist of the 13 protofilament, and I hope you remember we already discussed microtubules basically when they form the nanometer approximately 25 nanometer, and internal area, internal structure that is completely hollow. So we try to understand that is 25 nanometer structure, who helically all the that tubulin protein they helically arrange with each other, and after the arrangement they form a hollow structure. So we are taking what is meaning of the 13 protofilament, how they are showing helical arrangement, how they form the hollow structure, and how they form a 25 nanometer diameter. Chalo. Simply we are taking the structure. I'm using one outline. Suppose one alpha, one beta, one alpha, one beta, like this. So we are just draw a boundary. Alpha, beta, alpha, beta, alpha, beta. So all the tubulin proteins, they helically arrange with each other, and finally they form a structure like this. We already discussed this part, central part. Internal structure, it is completely hollow structure है, that is an empty structure. And if you are talking about what is the exactly diameter, so diameter basically it has 25 nanometer or 250 angstrom. So exactly these are the diameter. ठीक है, helically arranged है. Another one, what is meaning of the total 13 protofilament? So now if you see यहाँ पे आप बस दो feasible है. For example, these are the, suppose one bottle, एक water bottle imagine करो, boundary or wall of that bottle, these are the act as a boundary. Now you have to take one pen suppose ki bahut sare microtubules tubulin protein they attach with each other, so approximately 100, suppose 100 tubulin protein attached with each other, and now they form some elongated structure. So that pen you have to stick on the wall of your bottle. So one pen, two pen, three pen, four pen, total 13 pens they are arranged helically on the wall. Bottle a, internal area of the bottle is completely hollow, and whatever the wall is present on that wall, you have to stick tubulin protein but in a specific manner, in a helical manner. So kitne protofilament hai? Total 13 protofilament. It means what? Suppose one barrel hai, suppose one bottle hai, and on the that circle, suppose ये कुछ इस तरह से structure है, and on that structure now there is a presence of, suppose these are the 1, now 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So exactly all that around, all that circular area, completely that diameter, it is completely filled with the protofilament. And how many protofilament? Total 13. Okay, protofilament means what? When alpha beta they join with each other and they form a long elongated structure, that structure it is called as the protofilament. ऐसे कितने प्रोटोफिलामेंट होंगे? टोटल 1 3, टोटल 13. So one protofilament, then 2, 3, 4, they join in a circular manner, they join in a helical manner, and now that's why the internal area basically they form the hollow structure. The internal area they form the hollow structure who has the diameter approximately 25 nanometer. Got it? Clear? The boundary of the microtubules means what? Internal empty area hai. And now what happened? Whatever the walls are present on that wall, 13 protofilaments they get bind. And inside one protofilament, lots of tubulin proteins are present. Single नहीं होता, बहुत सारे होते हैं. It's depend upon GTP. If provide it GTP provide more, that time they are showing the more elongation. Simple. Okay, Komal? Clear? Okay. Yeah. Any doubt? Hmm. चलो एक सेकंड. चलो. Yeah. So after this tubulin structure, we are talking about the structure of the centriole. Hmm. So next part we are talking about centriole. Centriole, or you can say centrosome. Both these are the correct. So quickly I'm telling you, first of all, what is meaning of the centriole and where, what is the meaning of the centrosome and where it is exactly present. Suppose these are the one cell, ये एक cell है. and these are the animal cell. Hmm? Inside the animal cell, centrally located nucleus present है. So these are the nucleus. Now what happened? Exactly near to the nucleus, but in cytoplasm, यहाँ पे something, a body is present, proteinaceous body, like this dotted line. So this structure basically it is called as the structure of centrosome. Centrosome. And inside the centrosome, now there is a presence of pair of centriole. Just see, एक इस तरह से होगा, one, and now another one like this. ठीक है? And this color represent, that is pair of centriole, which is arranged perpendicular with each other. So these are the pair of centriole. Centriole which is present in pair. And basically arrangement perpendicular with each other. Like this. Simply ये इस तरह से, this one, and now these are the another second. So first and the second. So if you see 90-degree angle will be formed, and now these are the called as the which structure, that is centriole. So centrosome. Centrosome basically these are the one structure which is totally made up by the microtubules without plasma membrane structure, and now they are present inside the cytoplasm near nucleus but inside the cytoplasm. ये part important है because in next class ही बोल रही हूँ, in next chapter we'll start cell cycle, cell division. That time we have to focus on the centriole. So now centrosomes are present inside the centrosome. Inside the centrosome, pair of centriole is present. Pair of centriole. Again, centriole, these are totally made up by the microtubules without plasma membrane structure, and now arrangement, they are arranged in a perpendicular manner. Okay? So now quickly we are talking about some information about the centriole or the centrosome. Correct. So basically centriole, these are the without plasma membrane structure. First point, without plasma membrane. अब तक हमने देखा था ribosome without plasma membrane structure. Now we are talking about the second one, centriole without plasma membrane structure. Remember this point, okay? Centriole without plasma membrane structure, ribosome also without plasma membrane structure. Write down in your notebook margin. Another one, basically when we are talking about, they totally made up by the protein, that is microtubules. And you know very well, whenever we are talking about, whenever you are using microtubules, nothing but we are talking about they are made up by the protein. That name of the protein, that is tubulin protein. Number second. And the third point, कहाँ पे present होगा, question on the basis of their occurrence. Please remember, in case of animal, in case of the fungi, and in case of the algae, in these three organisms, typical centriole is present. But when you talk about higher plants like gymnosperm, angiosperm, so in case of the higher plant, such type of the centriole, it is totally absent. Question, remember this point. In case of the higher plant, in case of the gymnosperm, angiosperm, structure of centrosome or the structure of centriole, it is totally absent. Clear? Clear this point? Any doubt? Just quickly check. एक सेकंड दो. Yeah, okay. So these are the point where in case of animal, fungi, as well as in case of the algae, such type of the centriole present, but in case of the plant, it is absent. Now, another one, we are talking about typical structure, and now in this point, we are talking about arrangement 9+0 arrangement. So we are talking about structure of centriole. Okay? Structure of centriole. In case of the structure of centriole, basically they are specifically showing arrangement 9+0 arrangement. Question. 9+0 arrangement. So what is meaning of the 9+0? We'll try to understand this point. Basically, what happened when we are talking about the structure of the centriole, now it is look like a cartwheel. Cartwheel. So in a circular manner, basically. And in this point, there is a presence of the triplet fibril. Triplet fibrils are present, and such all triplet fibrils, they are showing joining by three microtubules. They join by with the help of the three microtubules. That is microtubules A, microtubules B, and the microtubule C. So we'll try to understand what is meaning of the triplet fibril, and they are joining by three microtubules. Suppose triplet means what? Total three. So ये हो गया one microtubule, that is A. Now on the A there is a joining of the B, second one microtubules, and finally on the B there is a joining for the C, that is C microtubule. So now this entire structure, these are represent as a triplet structure. This structure, it is called as the triplet fibril. Okay, so A, B, C, these are the three microtubules. Those are associated with the centriole structure, and A joined with B, B joined with C, and such typical structure after the joining they called as the triplet fibril. And please remember, in centriole, total total 9 triplet fibrils are present. How many? Total 9 triplet fibrils are present. So we try to understand with the help of this structure what is meaning of the 9+0, and how that triplet fibril they join with each other in helical manner, in a circular manner, so that's why it is look like a something a cartwheel like structure. Got it? So any doubt from the triplet fibril? Just try to understand. Triplet fibril means A, B, C, three microtubules say, and they join with each other, and after the joining, they can form a triplet fibril structure. Okay? Yeah. Any doubt? Any doubt? Hello? Okay, child. Yeah. Next part. Next part, we are taking the structure. I just draw some hypothetical boundary actually, so we'll get this some idea. And triplet. We have to draw total 9 compartment. 1, 2, 3, 4, 5, 6, 7. One, two, three, four, five, six, 7, 8, 9. Okay, total 9 compartment. Now, so they join like this. So total 9 कंपार्टमेंट, ठीक है. सेंटर में जो हमने व्हाइट कलर दिया हुआ है, these are called as center. It's called as hub. Now whatever the lines are there, these lines basically, which is given in the yellow color. So basically all these lines are called as the spoke. Spoke nothing but these are the some supportive structure, and total 9 spokes are present. One, if you count, total 9 spokes are present. Now यहाँ पे कुछ inside the notch again there is a something a protein supportive structure, because entire structure these are the proteinaceous. पूरा structure बना हुआ है. And now we are talking about triplet fibril, which is arranged. So just see A, B, C. Similarly, A, B, C. So A, B, C. So these are the total. Again, if you count A, B, C, total 9 sets are present. Now we are talking about the last point, that is linker. And name of linker, that is CA linker. CA linker means what? So C है. So quickly we are giving the name ABC, ABC. These are the 3 microtubules. So we are talking about the CA linker. C attached to A. This C, they are showing attachment with A. So this C, it is attached with the A. ये वाला C attach होगा with next A. This C attached with the next A. C, it is attached with the next A. Similarly, C, it is attached with the next A. C, A, and C, A, and finally that is CA. So now this typical structure name, these are called as CA linker, because C microtubule, they attach to the A microtubule of the next. So now what? These are the entire structure if you see. Number 1, triplet fibril presented. Number 2, 9 they are present towards periphery. 9 triplet fibril. Yes, कहाँ पे present है? They are present towards the periphery. They are present towards the surface. But if you discuss at the center, discuss at the hub also called as the center. So कितने present at the center? Only 0, because there is no any microtubule present at the center. So such kind of this arrangement, simply it is called as 9 plus 0 arrangement with respect to centriole. Triplet fibril, they present on the surface, they present towards the periphery. And now all this microfibril, because see plasma membrane nahi hai, there is no any boundary. All this structure they are made up by the simple protein, that is tubulin protein. So now what happened? बहुत कुछ होगा, like hub present, hub they provide extra support. Now CA linker present, all the microfibril they attach with the each other with the help of the CA linker, because now what happened, there is no any boundary, there is no any plasma membrane. So that's why such kind of the linker, such type of the spoke, they help to maintain structure. And 9 present toward the periphery and 0 microtubules they present at the center. That's why such kind of this arrangement, these are called as 9 plus 0 arrangement. Got it? Clear? Ramchandra, got it point? Don't worry, at the end of the class I'll share the whiteboard screen so you can complete your diagram. Yeah, any doubt from this point? दीपिका ठीक है. गार्गी 9 plus 0 arrangement. Remember centriole 9 plus 0. Yeah. Chalo. So quickly we are talking about functions. You know very well, basically they help in cell division because they formed spindle fiber. They form spindle fiber during cell division. So ये पार्ट हम देखेंगे आगे. They form spindle fiber during cell division. And the another important function, basically now what happened? The typical centriole, they help in formation of basal bodies. Please remember, okay, basal bodies. Only basal bodies, because जिस तरह से होता है ना कि flagellate is consist of the base, hook and the filament. Similarly, in case of the eukaryotic cilia and the flagella, base it is called as the basal body. So basal bodies of cilia and the flagella, and you know very well, cilia, flagella, basically they help for the locomotion. So please remember, entire structure में help नहीं करेगा. Centriole only they help to form only basal body, only base of cilia and base of the flagella they form with the help of the centriole. So these are the two functions with respect to centriole structure, and how they help for the spindle fiber formation. This part we will discuss in upcoming classes in chapter cell cycle, cell division. Chalo, got it? Hub means what? Center. It is made up by the simply protein. That hub means center. At the center, there is no any microtubule. Means like A microtubule, B microtubule, C microtubule. It is not present. Okay. This one, C. Clear? Acha, pink one is not visible. Okay, fine. Yeah. I'll change it. No problem. Zero means what? Zero means no microtubules. Okay? Zero means no microtubules. अभी हमने देखा ना, triplet microtubules are present. A, B, C, they present on the periphery. So A, B, C, one triplet fibril. So 1, 2, 3, 4, total 9 sets present. And at the center, zero microtubules. No A, no B, no C. That's why it is we are represent as a zero, because at the center only plain structure is present. Only protein is present, but not specifically A microtubule, B microtubule, and the C microtubule. Okay, that's why. Chalo. Now another point, cilia and the flagella. 9+2 arrangement. यहाँ पे confusion होता है. Next part we are talking about eukaryotic cilia and eukaryotic flagella. Okay, because in case of the bacteria, flagella present, but now here we are talking about eukaryotic cilia and the flagella. Very important part. When we are talking about cilia and the flagella, please remember plasma membrane is present. Eukaryotic cilia, flagella, plasma membrane is present. And the another protein, whenever we are talking about that cilia and the flagella, they are made up by the protein that is tubulin protein. So they made up by the protein that is tubulin protein. And the another one, for the movement, there is a requirement of ATP. पैसा चाहिए in the form of ATP. So for the movement, ATP is required. When you compare this point with prokaryotic flagella, because cilia तो होगा नहीं, obviously. So in case of the prokaryotic flagella, if you compare this point, now normally what happened? First point, these are without plasma membrane structure. Prokaryotic flagella without plasma membrane. Another one, they made up by the flagellin protein. Protein get vary, they made up by the flagellin protein. And number third, no paisa required, no ATP required for the movement. Got it? So these are the exactly major difference between the prokaryotic flagella and the eukaryotic flagella. Remember this point. Okay, plasma membrane present hoga with respect to eukaryotic flagella, plasma membrane absent with respect to prokaryotic flagella. Now That structure it is made up by the tubulin protein, and your prokaryotic flagella they made up by the flagellin protein. ATP required for the movement in case of the euflagella. In case of the proflagella, no requirement of ATP. Got it? Clear? Hmm. ठीक है, चलो. Now we are talking about, yeah. So we are talking about cilia as well as the flagella. Both they consist of three parts. They consist of the three parts. Number one, basal body. Number second, basal plate. And the number third, shaft. So these are the three parts. Those are associated with eukaryotic cilia and the flagella. But when you compare this point with prokaryotic flagella, so again prokaryotic flagella, they consist of the three parts, but simply it is called as the base. Then number second, that is hook, and the number third, that is filament. So structure may be vary hai, but functionally they are similar. So base or simply it is called as the basal plate, और नंबर सेकंड दैट इज हुक. आई होप यू रिमेंबर कुछ इस तरह से स्ट्रक्चर होता है. ये हो गया बेस. नाउ दिस स्ट्रक्चर इट इज कॉल्ड एज द हुक, and this structure basically it is called as the filament. Okay, so again prokaryotic flagella also they made up by the three parts, but parts they get vary: base, hook, filament, while eukaryotic flagella and the cilia they made up by the basal bodies, basal plate and the shaft. तो यहाँ से क्वेश्चन नहीं बनेगा consist of which part, so we are not taking those part. Okay, directly we are talking about structure of cilia and the flagella and what is meaning of 9+0 arrangement. नाम थोड़ा तो पता चल गया होगा. 9+0 means what? 9 present over the periphery and 2 present at the center. Microtubules. Hmm? Yeah, chalo. So now, next part we are talking about structure of cilia and flagella. And specifically they are showing arrangement 9+2 arrangement. So what is meaning of the 9+2? Before starting 9+2 arrangement, we try to understand one thing. Again, ये जो structure होगा, these are the cartwheel-like structure. All the tip proteins they are helically arranged. It's look like a circle. It's from a particular diameter. And now in this point, we are talking about there is a presence of doublet fibril. In case of the centriole, we discussed triplet fibril. यहाँ पे क्या present है? Doublet fibril. So now what happened? Doublet fibrils are present. And now these doublet fibril they consist of two microtubules. They consist of the two microtubules, that is microtubule A and the microtubule B. A microtubule and the another one that is B microtubule. So we try to understand structure. ये हो गया हमारा A को attach है B, and now this structure basically is called as the doublet fibril. One important point, just we discussed, for eukaryotic cilia and the flagella there is a requirement of energy in the form of ATP. So now what happened? There is a specific protein which is associated with fibril, doublet fibril. So यहाँ पे one specific protein is present, and now I am giving name for this protein. That protein basically is called as the dynein protein. I hope in zoology you know very well. Dynein protein basically these are the one specific type of the ATPase enzyme. It having ATPase activity, and you know very well if we are talking about the ATPase activity, it means that ATP undergo the hydrolysis, they convert into ADP plus inorganic phosphate. And now what happen? Whatever the energy it get released, that energy it is used by cilia and the flagella for the movement. Okay, so now what happens specifically? We are talking about there is a presence of one specific type of the protein, dynein protein. Dynein protein itself, it has the ATPase activity, and with the help of the ATPase activity, ATP converted into ADP plus inorganic phosphate, energy get released, and once there is an energy get released, now finally they can form cilia, means that energy it is uptake by the cilia and the flagella movement. So remember, doublet fibril होंगे, and now that doublet fibril they having one specific type of the activity, ATPase activity, because dynein protein attached to microtubule. Got it? Yeah, clear? Any doubt from the doublet fibril, doublet and the triplet? Just try to understand. Okay, doublet and the triplet fibril. Triplet होगा for the centriole, doublet we are talking about for the cilia and the flagella. वहां पे था 9+0 अरेंजमेंट, हियर वी आर टॉकिंग अबाउट 9+2 अरेंजमेंट. 9 present towards the periphery and 2 present at the center. 9+2 arrangement. Got it? Achar? Okay. Yeah. Any doubt? Any doubt? ठीक है, चलो. सो इसका स्ट्रक्चर देखते हैं थोड़ा सा. क्वाइट कॉम्प्लिकेटेड है. Complicated in the sense, lots of names actually get involved. That's why. Simply I draw a hypothetical boundary because it has the plasma membrane. So these are the outermost boundary, that is plasma membrane, because cilia, flagella, plasma membrane present. Now we are talking about the another structure. First of all हम start करें from the center. ठीक है, 9+2 है, so we have to draw two microtubule which is present at the center. So यहाँ पे देखो at the center, which is represented by this orange color. Okay, such structure which is represented by this orange color, simply these are called as central exoneme. This structure it is called as the central exoneme. So now tell me how many central exonemes are present? 1 and 2. So now at the central exonemes are present, and please remember central exonemes, these are the nothing but these are the specific type of the microtubules. Those are present. So singlet present है, single में present है, right? So the central exoneme basically they present in a singlet form. सिंगल प्रेजेंट है, डबल नहीं यहाँ पे, ना ही ट्रिपल है. 1 and 2, right? That is central exoneme. Now that central exoneme simply they join with each other like this. तो यहाँ पे joining होगा. Now we are talking about that central exoneme, which is a specific type of the microtubule which is present in a singlet form. They protected with some sheath, and that sheath called as the central sheath, like this. Okay, so ये क्या है basically? These are called as the central, because central exoneme and central exoneme is covered by this some protective layer. That protective layer simply we are talking about central sheath involved into protection. ये भी protein से बना हुआ है. Now next part we are talking about, there is a presence of doublet fibril which is present on the periphery, and you know very well how many sets are present. 9+2 means total they are present in a 9 set. So we try to draw those structure. Just sorry. हाँ मेरी माँ, थैंक यू। कहाँ थे हम? या डुबलेट फॉर्म। सो डुबलेट फॉर्म 1 2 टोटल 9 सेट। सो ये था 1 2, तो ये हो गया A and B, and you know very well A and B basically they having dynein protein, तो वो भी हमें ड्रॉ करना पड़ेगा like this. So there is a dynein protein. ठीक है, चलो सो क्विकली टोटल 9 सेट ड्रॉ करो। 9 सेट, 2 3 4 5 6 7 8 and the 9. A B डुबलेट फॉर्म में प्रेजेंट है। And again there is a presence of the dynein protein who has ATPase activity. ठीक है, now so just see ये जो हमने ड्रॉ किया, this one basically simply it is called as the peripheral exoneme. And again this peripheral exoneme which is made up by the protein which is made up by the microtubules. So again they made up by the empty, that is microtubules, but now they are present in which form? They are present in a doublet form. It is not present in a singlet. तो doublet form में present है. Again they consist of the dynein protein also. ठीक है. Now what happened? We are talking about the spokes because protection तो चाहिए ना. So next part, that is spokes. One spoke, two spoke, 3 1, 4 1, 5th one, 6 1, 7, 8, and finally 9. So how many spokes are present? Total 9 spokes are present. So this color, these are called as the spoke who provide support. ठीक है, so just see सेंट्रल एक्सोनीम हो गया, पेरिफेरल एक्सोनीम हो गया. सेंट्रल एक्सोनीम टोटल 9, सॉरी सेंट्रल एक्सोनीम टोटल 2 एंड पेरिफेरल टोटल 9. सो 9 + 2 अरेंजमेंट, बाकी का सब कुछ सपोर्टिव स्ट्रक्चर. प्लाज्मा मेम्ब्रेन प्रेजेंट है, स्पोक्स आल्सो प्रेजेंट. लास्ट पॉइंट वी आर टॉकिंग अबाउट लिंकर, दैट इज AB लिंकर. Last point, AB linker. So A attached with the B like this. So this structure basically is called as the AB linker. So this structure, finally we are talking about the linker. Name of the linker, AB linker. That's it. So such structure template is called as the 9 + 0 arrangement, sorry 9 + 2 arrangement, because 9 present towards the periphery, and that structure basically it is called as the peripheral exoneme, and 2 present at the center in a singlet form. That's why it is called as the central exoneme. Okay, that's it. So these are the total entire structure with respect to 9 + 2 arrangement. I am telling you diagram के ऊपर question नहीं बनेगा. Question directly they ask from the 9 + 0 and the 9 + 2. So don't waste your time to draw the diagram. I'll share the whiteboard screen. जब आपको time होगा आप तब से draw करो. That time you can draw. Just 1 second, okay? Okay, should I continue? Yeah, diagram done. So these are the simple structure with respect to doublet fibril, showing the 9+2 arrangement. 9 doublet fibril, they present toward the periphery, that is peripheral axoneme, and 2 present at the center. Simply it is called as the central axoneme. Clear? Yeah. So next point, we are taking some question and you have to tell me the answer. Yeah. So one point, suppose question is that how many microtubules, how many microtubules are associated with number of the microtubules, okay? How many microtubules they are associated with? Number one, centriole, and the number second with cilia and the flagella. Okay, I'm giving one hint. पहले हमने देखा था they are showing the arrangement, that is 9+0 arrangement, and now they are showing the 9+2 arrangement. ये जो 9 present है, now they present in a triplet. Now such one 9 they present in a doublet form, and this 2 which is present at the center, that is present in a singlet form, right? So now tell me the number of the microtubules. How many microtubules they are associated with structure of centriole and the structure of cilia and the flagella. So give me just 2 minutes, you can solve this question. I'll come back. Yeah, okay, chalo, answer आ गए. So let me check. 27 microtubules for the centriole and the 20 microtubules for the cilia flagella. Very good. Yeah, answer is correct. Because now what happened? we'll discuss that is triplet form, right? It means that is 9 into 3, total 27 microtubules. Those are associated with structure of centriole. And now in this condition, doublet hai, matlab 9 plus 2, that is 18, and singlet plus 2. So total basically 20 microtubules, they are associated with structure of cilia and the flagella. Okay, yeah, that's it. So if you solve module, not module, basically assignment, तो आपको assignment के एक question मिलेगा. Calculate number of the microtubules, those are associated with the centriole and the cilia flagella. ठीक है? Yeah, got it? So next point we are talking about, in case of, अब तक हमने देखा, just quickly we are revising all these part. Right now we'll discuss about, there is a presence of the two organelle, number one centriole and the another one that is ribosome, both type of the ribosome. Okay, 70s ribosome and the 80s ribosome. Basically these two organelle, they are non-membranous organelle, non-membrane organelle, without membrane organelle. Okay, now when we discuss about, in case of the plant, in case of the plant, centriole is absent. In case of the plant, centriole is absent, right? But ribosome is present. And which type of the ribosome? 70s ribosome also present, as well as the 80s type of the ribosome is present, because 70s type of the ribosome, these are associated with structure of the chloroplast. Even it is associated with structure of the mitochondria. So centriole absent, but ribosome is present. Now what happened? When you compare this point with the animal, in case of the animals, dono present, that is centriole also present, and as well as structure of typical ribosome, that structure also present. And in case of the ribosome, similarly 70s type of the ribosome is present, as well as there is a presence of the 80s type of the ribosome. But when you compare this point with bacteria, so in case of the bacteria, only ribosome is present, because centriole, these are the typical structure which is associated with the eukaryotic organelle. So centriole absent hoga. But in this condition, in case of bacteria, ribosome is present, and you know very well, 80s type of the ribosome, those are associated with structure of bacteria. So these are some basic extra information with respect to centriole, as well as the ribosome, which is a without membrane organelle present in plant, animal and the bacteria. Means presence and absence. Ribosome to sabhi mein present hoga, but centriole absent in bacteria, as well as it is absent in higher plants. Got it? Yeah. Clear this point? Clear? चलो Now, last point of this chapter, last organelle, double membrane organelle, brain of the cell, nucleus. So any doubt from the cilia, centriole, centrosome, any doubt, you have to revise this part. ठीक है, आज हो गया तो आज इसको cover करो. Hardly take 30 minutes. Within 30 minutes, revise each and every part. Try to read notebook first. I'm telling you, don't go NCERT. First of all, आपको notebook study करना है. That is your own class notebooks. Then go through with the NCERT. Then you can read module and immediately you can solve questions also, because already assignment shared. आपको DPP मिल रहा है. I think you got DPP yesterday. Today also you will got one by one DPPs. So try to solve it. Okay. Yeah. So please don't go NCERT first. First read notebook and then secondly you go through NCERT. ठीक है. Yeah. So next part we are talking about brain of the cell, last organelle, nucleus. You know very well, first time scientist Robert Brown discovered the nucleus from the orchid root cell, and even that nucleus term is coined by the same scientist, that is scientist Robert Brown. तो जितना कुछ nucleus के related coin term by or the discovery, all this contribution given to scientist Robert Brown. Yeah, screen is visible. Pratyusha, screen is visible. Yeah. So now you know very well, when we are talking about the cell may be uninucleated, cell may be multinucleated, depend upon the nucleus. So any cell, basically they are uninucleated, or maybe they are multinucleated. Normally what happen, such multinucleated condition, they again divide into two types. What kind of the multinucleated? Number one, we are talking about, that is syncytium. Syncytium. And syncytium condition, simple meaning, many cells they get joined. Many cells get joined and now they formed a multinucleated condition. They form multinucleated. So initially what happens, simply we are talking about fusion of the cell. Many cells they get joined and now they form multinucleated condition. Such condition simply it is called as the syncytium condition. Another condition होता है sinusotic condition. Sinusotic. In case of the fungus condition, we already discussed sinusotic, nothing but inside the cell, during the cell division, only and only karyokinesis will be occur, no cytokinesis. Only which condition is present? Only karyokinesis, and in this condition, there is a no cytokinesis, no cytoplasmic division. And now what happened? Finally, whatever the cell will get formed, so now that cell basically it is formed the multinucleated common cytoplasm, and inside the cytoplasm many nuclei are present. Why? Because karyokinesis is present but there is a no cytokinesis. So that's why such condition they leads to formation of the sinusotic condition, multinucleated condition. So depending, during the cell division they can naturally form, or now what happen, many cells they get joined, they form a common boundary and finally they are become a multinucleated. Suppose like these are the one cell, another cell, third cell, fourth cell, fifth cell. Each and every cell they are having nucleus. Eventually they get joined, and after the joining, now what happened, they can form a common cell, and inside the cell now many nuclei present like this. So such condition simply it is called as syncytium condition. Okay, syncytium, simple condition. So dependent, but both these are the part of the multinucleated. Many cells they can join and now they converted into multinucleated, or during the cell division, cell undergo only karyokinesis, cell undergo only nuclear division, no cytokinesis, no cytoplasmic division. That time also they can lead formation of the multinucleated cell. Okay, yeah, clear? Any doubt? So you know very well, nucleus basically it is absent in prokaryotic cell. It is absent in mature mammalian RBC. Even it is absent in seeds tube element in case of the plant. While nucleus is present in rest of all eukaryotic cell. So it is absent in seeds tube, absent in RBC, absent in prokaryotic cell. तो वो part हम नहीं देख रहे because that is very basic. Next part. Yeah. Structure of nucleus. ठीक है? Structure of nucleus. Basically, inside the structure of nucleus, we'll talk about the total 4 part. Number 1, we are talking about nuclear envelope. First one. Number 2, nucleoplasm. Number 3, chromatin. That is genetic material actually, but present in chromatin fiber. And finally, without membrane structure, dark body, that is nucleolus. So in this point, we'll discuss the total 4 part: nuclear envelope, nucleoplasm, chromatin structure, and as well as there is a presence of the structure of the nucleolus. Okay? Now, first of all, we are talking about the nuclear envelope. Number 1. Chota hai, quickly we finish this part. Nuclear envelope. Basically, nuclear envelope, when we are talking about the nuclear envelope, basically they consist of the 2 membrane, outer membrane and the inner membrane, because nucleus it is a double membrane organelle. So it has the 2 membrane, outer membrane, and as well as we are talking about that is inner membrane. Okay? Now when we are talking about the outer membrane, such outer membrane, so first of all we are talking about the outer membrane. So I hope you remember लास्ट क्लास ऑल्सो व्हेन वी टॉक अबाउट द एंडोप्लाज्मिक रेटिकुलम, हमने एक पॉइंट डिस्कस किया था. आउटर मेम्ब्रेन दे आर शोइंग एक्सटेंशन एंड नाउ दे कैन प्रोवाइड, they form a rough endoplasmic reticulum because they provide the more surface area, and finally the ribosome they can attach, and now with the help of the protein synthesis can be occur. So whenever we are talking about the outer membrane basically they can form number 1 rough endoplasmic reticulum, and as well as they can provide attachment site for the ribosome. So both rough ER as well as ribosome, they are showing attachment. Form kene sacha, we are talking about outer membrane. With outer membrane, there is an association of 2 organelles, ribosome as well as the rough endoplasmic reticulum. While when we are talking about the inner membrane, because inner membrane is not showing the extension, they are not having the means they are not having this typical structure. So now that point of view, that inner membrane basically they help to form formation of the smooth structure. So they can form a smooth structure, means they can form a boundary, innermost boundary. Now between the nuclear membrane, means between the outer and the inner, suppose ki ye membrane hai, these are the entire membrane, outer and the inner. Okay? Now through the membrane there is a small gap is present, and that gap nothing but, these are the something a gap is present, and that gap nothing but these are called as nuclear pore complex, NPC. Simply these are called as the nuclear pore complex. So if I'm talking about nuclear pore complex, because basically lots of, means millions of nuclear pore complex they are associated with the membrane. Suppose a outer membrane, these are the inner membrane, and the outer and the inner membrane simply it is crossed by some gap, they cross by the simply pore. That pore basically it is called as, simply they are called as the, that is nuclear pore complex. Okay? Now when we are talking about the nuclear pore complex, basically these are the site where proteins, enzyme, even ribosomal unit, messenger RNA, they are showing movement from site. मतलब exactly क्या करे, these are the bidirectional, means one movement they can, molecule they can go outside through this pore complex, and again they can come inside also. So simply what happened, there is a movement. But this nuclear pore complex, a very important point, please remember, these are the highly selective in nature. Pore है definitely, but it is not allowed each and every molecule. They allowed only those molecules those are related with nucleus, like for example messenger RNA form inside the nucleus, so that messenger RNA they can go inside the cytoplasm via that pore. मान लो कि histone protein है, you know very well, protein synthesized in cytoplasm, so that histone protein they can enter through that nuclear pore and they enter inside the nucleus. So now what happened, there is an exchange of material they are go inside, they can come inside as well as they go outside. So this nuclear pore complex, millions of nuclear pore complex present on the nucleus plasma membrane, but they are highly specific. अगर काम होगा, that if that part, if that enzyme protein that related to nucleus, that molecule also they get entered, otherwise there is no entry for other molecules. So histone protein they can enter, messenger RNA they can enter exit, some enzymes they can enter exit, lamin protein हो गया, chromatin हो गया, so whatever replication के लिए enzyme चाहिए, for the transcription enzymes are required, for capping 5 prime capping, 3 prime adenylation, splicing, बहुत सारे process होते हैं. So for the replication, for transcription, whatever material it is required, all that material they can exit and they can enter in nucleus via this nuclear pore complex. But it's highly specific. Got it? Got it? Komal, finally both look same, no? Who? You and me? Who looks same? You and me? Okay, Ghislaine. Komal. Huh. At the end, they look same. Yeah. Sinusoidal condition and essential condition, both they look same. Yeah, both they are multinucleate, but किस तरह से form हो रहा है? What is the pathway for the formation? That get vary. Okay, yeah. So now with this we end today's session. Next class हम diagrammatic representation करेंगे with lamin protein, chromatin. Then some point we'll discuss how the chromosome structure, euchromatin, heterochromatin, and another important point, satellite DNA वो discuss करेंगे, and the nucleolus organelle, because nucleolus these are the responsible for formation of the rRNA, ribosomal RNA. So ये जो part है we'll discuss in next class, and again from the next class we start the next chapter, cell cycle and the cell division. Okay, yeah. So any doubt from today's class? Any doubt?